In this video, we will discuss about what is the proposed N-Cadherin and MMP9 model that this paper finally proposed based on the results that they have observed from their experiments. So, what they said is that this PMA will trigger this MAPK or MAPK pathways, and this MAPK pathways thereby leads to the expression of this MMP9 in the cellular matrix, and this MMP9 will then cleaved off this N-terminal side of the cadherin, which is expressed on the cellular side, and this N-terminal fragments is usually around 95 kilodalton. Simultaneously, it will also enter inside this. C-cadherin intercellular part of this cells, where it will cleaved off this C-terminal fragment, and it will cleave off two different kind of C-terminal fragment. That is one of 40 kilodalton and second one is of 35 kilodalton. However, this C-terminal fragment cleavage can be prevented by using this secretase inhibitor. And when they use secretase inhibitors, only these C-terminal 2 fragments will be formed, and it will translocate into the nucleus. And once it is however, how this CTF2 after cleavage translocated into nucleus is not yet known. That is why the question mark is there. So once it is translocated into the nucleus, it will lead to the positive autoregulation of this MMP9, and this MMP9 will then overexpressed. So basically, this is a cascade effect. Once the MMP9 are formed, it will cause translocation of this C-terminal fragment 2 of the N-Cadherin, and it will further upregulate or autoregulate this MMP9, and this overexpression of this MMP9 thereby leads to the excessive cleavage of the N-Cadherin, both at N-terminal side and also at the C-terminal side. And this C-terminal sides cleavage leads to the CTF2 formations, since the CTF1 cleavage has been prevented by these secretase inhibitors. And this CTF2, once it is translocated into the nucleus, then it will cause once the excessive CTF2 is translocated into the nucleus, then it will cause this increased invasions. And this is based on all the results that we have also discussed that are presented in their main figure, that shows that these PMA treated cells along with these secretase inhibitors will cause increased invasion. At the same time, from the immunofluorescence we have seen, it will also leads to this CTF2 overexpressions, and this is a model that is been proposed by this paper finally. And in this case, another thing that one need to understand is about how the CTF2 is translocated into the nucleus. They just mentioned that they are able to just see this CTF2 overexpressions in the presence of secretase inhibitors. However, when they see it in the Western blot, they said that because of the overexpression of this CTF1, once it is its cleavage is prevented, they are not able to see CTF2 formations, and that things they have validated using this GFP conjugated CTF2, and but the mechanisms how this CTF2 is translocated into the nucleus, whether it makes a complex with something else or it translocated on its own to the nucleus, is not being studied in this paper. And one more thing that this paper does not answer is this the level of positive autoregulations. They did not quantify how much what is the amount by which it will be positively autoregulated once this cleavage starts to occur. They just mentioned the relationship that once they use the secretase inhibitors that prevents the CTF1 cleavage, it will cause the CTF2 to translocate, and that will lead to the overexpression of this MMP9 and the excessive cleavage of the N-Cadherin, both at the N-terminal site and also at the C-terminal site. If the secretase inhibitor is there, then it will cleaved at the CTF, cleave to make or to form CTF2. This is the model that this paper proposed. I hope you understood this model and the results that we have discussed about this N-Cadherin and MMP9 relations, or how they are related to each other. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.